My name is Vaidehi Goyal. Did you know that there are certain worms that eat plastics? Well, my project is about the effect of mealworms on plastic decomposition. The purpose of this experiment was to determine if mealworms could consume a polystyrene or a styrofoam, a high-density polyethylene or a milk jug, and a low-density polyethylene plastic or bread bag. Why is this research important? Plastics can cause contamination in natural habitats such as the ocean. Animals can consume those plastics mistaking it for food, which can lead to two things. First, carcinogens in the plastics can cause cancer in the animals, therefore endangering their species. Secondly, the plastic now enters a long food chain, starting from the animal, eventually ending up in the human. How can we fix this problem? Well, mealworms may be a solution. Mealworms consume plastics, and the only byproduct is a small amount of carbon dioxide. My hypothesis for this project was that if the mealworms were given a polystyrene, a high-density polyethylene, and a low-density polyethylene plastic, then the mealworms would be more likely to consume the polystyrene plastic. Here's how I did my project. I started by taking 3 different types of plastics: a bread bag, or a low-density polyethylene, an egg carton, or a polystyrene, and a high-density polyethylene, or a milk jug. I then cut out 25 equal-sized pieces of each of the plastics and recorded their weights. I placed each of the plastics into separate containers with 4 mealworms each, closed the containers, and left them for 2 weeks. After the 14 days were up, I then remeasured the weights of the plastics. These were the results. The mean of the styrofoam compared to the mean of the milk jug indicated that the mealworms had no impact on the milk jug's decomposition. And a t-test told us that this data was significant because both the means were different. The mean of the styrofoam compared to the mean of the bread bag indicated that the mealworms did have an impact on the bread bag's decomposition. A t-test told us that this data was insignificant because both the means were the same. Lastly, the mean of the bread bag compared to the mean of the milk jug indicated that the mealworms had a larger impact on the bread bag's decomposition. And a t-test told us that this data was significant because both the means were different. Due to these results, the null hypothesis was rejected and the research hypothesis was supported. All t-tests were conducted at a 0.05 accuracy level and had a degrees of freedom of 48. This table represents the statistical analysis of my project. This was my raw data table, and this is the graph representing the information and data of my project. In conclusion, the mealworms were able to consume the bread bag and the styrofoam, but were not able to consume the milk jug. The mealworms were able to consume the polystyrene, or the styrofoam, due to a bacterium found in their gut called Exiguobacterium. This bacterium may not have been helpful in breaking down polyethylene plastics. Waxworms, which are worms who break down polyethylene plastics, contain 2 main bacteriums: Enterobacter asperi and Bacillus. Mealworms may not contain these 2 bacteriums, therefore making it harder for them to break down polyethylene plastics, such as the milk jug. Another explanation may be that the milk jug was too hard and too dense, or too crystalline, for the mealworms to chew through. This project was obviously not perfect, and there are a couple of sources of error. First, the mealworms were only given 14 days to consume the plastics. A longer time period may have resulted in better or even different results. Secondly, only 4 mealworms were placed into each container. A larger number of mealworms may have also resulted in different or better results. For further study, the effect of waxworms on polyethylene decomposition could be studied, or the effect of antibiotics on the mealworm's ability to consume plastic could also be studied. Thank you for listening.